hello everybody, and welcome to our webinar today. Today we'll be talking about cutting-edge approaches for cancer target discovery and validation, and we'll be joined by Wayne Doyle from Eclipsebio and Michael Garbati from Active Motif. They will be giving each their presentation, and then we'll be taking Q&A after. So please enter your questions into the Q&A function at the bottom of your screen, and then we'll address as many of those as we can at the end of the talks. If you might mention who your question is posed to, if it's specific. And with that, we'll get right into it. We'll be getting started with Wayne Doyle, who's head of platform at Eclipsebio, and his topic will be powering cancer drug discovery with RNA and NGS from targets to validation. So Wayne, over to you.
Great, thank you very much. Let me just share my window. I'm excited to talk to you all today about how at Eclipsebio we can help power both cancer research and discovering different types of therapeutics that can be used for very effective cancer treatments. So to start, I just want to give a brief introduction to who we are at Eclipsebio. And at Eclipse, really our mission is to make sure that our partners' RNA research and drug discovery is a success. And we do this by developing different types of next-generation sequencing assays, and then provide the data resulting from that from our comprehensive bioinformatics workflows to reveal different aspects of RNA biology. And we provide support in three main areas for basic research, so determining what's actually happening with an RNA, how's it folding, what is binding to it, but then also target discovery and drug validation, which I'm going to focus on today. And then in terms of who we help, we've helped 24 of the top 30 biopharma, as well as multiple biotechs and multiple academics to achieve their research and drug discovery goals. But today I'm going to focus specifically on how we provide support in the area of cancer biology and drug discovery. So cancer is a devastating disease. There's unfortunately over 2 million new cases every year and over 600,000 deaths a year as well. And in terms of how cancer is often treated, there'll typically be a protein-centric approach. So a protein will be identified that is potentially leading to tumorigenesis, and then a therapeutic will be developed, such as a small molecule. That small molecule will bind to a druggable pocket, prevent that protein from having its activity or changing the activity of that protein. The issue with this approach is that only about 10% of proteins are actually druggable. And so this really limits the amount of space in which you can actually develop an effective therapeutic to treat a cancer. And then in addition, the majority of small molecule drugs will actually fail once they enter clinical trials. And so this makes it important that there are different approaches that can expand the scope and allow for new modalities to treat cancers. And one of the approaches that can be taken is, rather than taking a protein-first approach, is to instead target RNA. And one of the benefits of this is that RNA encodes all protein coding genes, so you're not limited to just the 10% of proteins that can be drugged. You can now drug anyone that's implicated in cancer, as well as targeting any long non-coding RNA. So there are RNAs that also don't produce protein, but are very involved in disease progression, and those can now be therapeutic targets as well. And when we talk about different ways of drugging RNA, there's really three main approaches. The first is similar to what's done with proteins. So you develop a small molecule. RNAs are very complex molecules. They'll fold into different secondary and tertiary structures. That creates druggable pockets these molecules can come into and change the activity. But then there's also different approaches based off RNA being a nucleic acid sequence. Since it is a nucleic acid, it can pair with other sequences. And so this underlies things like siRNAs that use RNA interference. You develop a short RNA, it brings the RISC complex to the target and degrading the target RNA, as well as ASOs, which can enable the degradation of RNAs or change activities such as changing splicing so you have the therapeutic effect. And so these are all RNA targeting approaches, and then what I'm going to focus on today. But I did want to just briefly mention that there are also approaches for cancer treatments that actually use the RNA as the therapy. In this case, it's a personalized cancer vaccine approach, and you determine what protein is uniquely expressed within a cancer or tumor. There'd be an antigen that could activate the immune system. You then produce that as an RNA, package it into an LNP, and deliver it to a patient. That then triggers the immune activation, and the patient's own immune cells can then go after the tumor, go after the cancer, leading to remission. So that is another powerful approach using RNA, but not one I'm going to really focus on today, but instead touch more upon how you can discover the ideal targets for one of these small molecule siRNA or ASO approaches. And when developing these types of therapies, there's really two main steps that need to be accomplished. The first is figuring out what actually to target, and then once you know what RNA you want to target, what region of it should actually a drug be developed against. And then once you develop the drug, you also need to validate it. Is it having the expected effect, or is it leading to off-target effects that could limit a clinical trial? And so for today, I'm just going to go through two short vignettes touching on each of these two topics. First, starting on the area of target discovery. And this is actually a very complicated problem. We often think about RNA as just a string that's floating in a cell, but the biology of RNAs is actually very complex, and there's multiple dimensions that need to be examined to ensure that you will have a very effective therapeutic once it enters the clinic. What I'm going to focus on today is that you need to understand what actually isoform of your target is expressed, if there's unique UTRs, which are the untranslated regions at the 5' and 3' end of a transcript. I'm going to go more into this on the next few slides. But in addition to knowing actually what transcript is present, you also need to know, is there structure that's going to be a problem? An RNA will fold into different conformations. This can create a druggable pocket that can be good for a small molecule. On the other hand, some structures can be prohibitive, prevent an siRNA from binding, prevent an ASO from binding. It's also key to look at how well is this RNA being translated. Ribosomes are very large complexes. They move very quickly. They unwind RNAs. And so they can act as competition for your therapeutic, limiting its overall efficacy. And then also in the area of competition, any RNA in a cell is going to be bound by proteins. These include regulatory proteins that bind directly to RNA, but also the RISC complex that's targeted using microRNAs. These can be competition for a therapeutic. You don't want to target something that the large protein complex is already at. But on the other hand, there can be situations that you want to disrupt that binding. That's often done with ASOs, some of the successful ones that are currently in the clinic, as well as anti-mirror approaches that attempt to disrupt microRNA binding. And so knowing where this binding patterns are occurring can help you develop a very effective therapeutic on knowing where to or not target. And then finally, RNA can also have base modifications such as methylation, and this can change the ability of a therapeutic to bind. And so mapping that can give you a full picture of where should I target or where should I not target. For today, I'm just going to focus specifically looking at UTRs. Again, these untranslated regions at the 5' and 3' end of a transcript. And the reason that I want to focus on these today is that they do provide a lot of unique information for cancer biology itself. Different cancers will have very specific isoforms that are expressed that can lead to the actual tumorigenesis. And so being able to uniquely map what those isoforms are gives us a deeper understanding of both the biology, but then also reveals where is a unique druggable region. I'll show this more in the next few slides, but once you know this is the exact isoform that's in a cancer, versus the isoform that is present in a normal tissue, you can look at what is a sequence that's only present in the cancer to make sure that if you target that, you won't affect normal processes and the patient can stay healthy. And then finally, for UTRs, these are also key regulatory features. They guide stability, they guide translation. And due to that, they often have high amounts of protein binding and microRNA binding, and these can be causative for cancers. And so being able to know which UTR is present, what proteins could be binding on that, can open new avenues for drug discovery, develop an ASO to prevent that binding or prevent that microRNA from having activity. And so in order to look at these UTRs, there's a number of different approaches. You can look just in a genome annotation, but each gene typically has multiple different UTRs labeled. It's not clear which one is actually being active in the cancer. You can do RNA sequencing, but that's typically very broad reads. It's hard to pinpoint exactly where does the UTR start or end. And so to overcome these limitations, we have developed our RNA-Seq suite of assays. These directly reveal with pinpoint resolution where does a UTR start or where does it end. And we have two flavors of this. The first is 5' N-seq. In this case, we enrich RNA starting directly from the 5' cap, and then we create sequencing libraries that tell us this is the exact base position where the 5' UTR starts on every expressed gene in a cell line or in a tissue. Similarly, our 3' N-seq is a complement. It enriches RNA from the poly A tail, and so then we can say this is exactly where the 3' UTR ends, right before the tail begins. Again, base level positioning of where the UTR ends are located from both the 5' to the 3' end. And so just to show an example of the type of data that's provided from this, I have data that Ribometrics, a drug development company, performed with us. This is from their poster that they did at last year's AACR. And they looked to see what isoforms of c-Myc are present within cancer. I'm showing here, this is the N-seq data. So this is next generation sequencing data where the height is the amount of sequencing reads at a given position. As you can see, there's only two very strong peaks. We don't see much reads occurring anywhere else. And so these are the exact bases where two isoforms are being produced within this cancer. There's a short version and then a little bit longer version that's present. And so now that we have this information, Ribometrics was able to go in and say, we want to reduce the level of c-Myc RNA within this cancer. We know that there's two isoforms present. So targeting the long isoform would not be productive because you'd still have the short remain, potentially still leading to the generation of the cancer. And so they decided to target this region that's going to be unique across all different isoforms. So when you have a therapeutic that targets that, all will decrease and you can reduce the level of the cancer or even enter it into remission. Once they identified this region, they then did structure profiling and looked at what's the actual secondary structure of this RNA within the cancer cells, and then found a region that would be an ideal druggable target and developed two different therapeutics that can both bind to this region based off its structure and the uniqueness of the sequence. And those two therapeutics, RBXG and RBXS, and they tested them in a cell line, a lung cancer cell line with high levels of c-Myc, and they found that in both cases it reduced the amount of c-Myc that was present, as well as reducing downstream targets of MYC as well. And importantly, this was very selective therapeutics. They also tested in a cell line that didn't have c-Myc, but instead had N-MYC, and both of these therapies had no effect on N-MYC levels, indicating it will be specific to just the cancer, allowing for very effective potential treatments in the future. So that was just one example of how looking at UTRs can reveal what are a specific region that should be targeted with a therapeutic, enabling kind of an effective design. But as I mentioned earlier, this is just one dimension of all the dimensions that need to be examined to really ensure that your therapeutic will be as effective as possible, that you're targeting the ideal site. And so in order to support our partners, we actually have a suite of technologies that reveal information on each of these aspects, such as our E-shape assay that directly measures the secondary structure of RNA within cells, or our RBP-eClip and MIR-eClip assays that map protein and microRNA binding respectively. But for now, I'm going to move out of the target discovery space. Once you've used this information to develop exactly where it should be targeted and then designed a therapeutic, we can then also validate that it's having the expected activity. In order to validate that a therapeutic's actually having what you want it to do, there's three main aspects that need to be examined. The first is just to make sure, is your therapeutic actually binding? You've designed it to target a specific site, but different things happen in cells. There can be competition that wasn't caught, different components. And so it's important to confirm, is it actually binding? And then once you've confirmed the binding, it's also important to make sure, are there potential on-target effects? It's bound to your target RNA. Is it doing what you want it to do? And then are there also off-target effects? Is it changing the expression of a gene that could be detrimental for a clinical trial or lead to a loss of patient compliance due to side effects occurring? And so it's important to examine all three of these components. For today, I'm just going to focus specifically at looking at on-target and off-target effects. And when we look at the on-target and off-target effects, I want to take a step back and talk about really what dimensions that should be. We'll often think about the effect of a therapeutic targeting RNA on just the levels of the RNA. So a therapeutic would bind, lead to degradation of the transcript, and then no protein is made. But when we're targeting RNA, there's actually two different areas that can be affected. It can change the level of the RNA. Alternatively, the RNA can stay there, but you've changed translation. So preventing the translation from occurring and then preventing the protein from being produced. And so this makes it critical that when developing a therapeutic that targets RNA, that you don't just examine the level of the RNA, but also examine the level of translation to get a full picture of how that therapeutic acts on both the target and off-target genes. And so we support our partners in doing this by using a optimized ribosome profiling assay that we've developed called eRiboPro. In this assay, we take a sample that has some RNAs that are being translated, some RNAs that aren't, and then we split this sample in half. For half of it, we extract out the RNA and just do a typical RNA sequencing library. So this will show when your therapeutic is present, are there specific genes that are increasing their expression, decreasing their expression? Are there networks of genes that are changing that can be beneficial or problematic? But for the other half of the sample, we lock the ribosomes in place, and then we digest all the free RNA. So the only RNA that's going to be left is directly within the ribosome complex. And then this gives us nucleotide level positioning of where a ribosome's acting, revealing really the dynamics of translation and the amount of translation that's occurring. Which then after we do next generation sequencing, we can do a variety of analyses, including looking similar to gene expression. Are there specific genes that have increased translation, decreased translation, or are there whole networks of genes that their translation is changing overall? And this change in translation is what I'm going to focus on for the remainder of the talk. And when we talk about changes in translation, really there's two approaches that are typically done, especially if you're trying to actively change translation as opposed to it being an off-target effect. One is that you're just looking to prevent translation. You don't want the protein to be produced, and so you want to prevent ribosomes from associating. You want to prevent translation initiation from occurring. The second approach is to actually slow down translation. So you allow ribosomes to associate, they'll start moving along the transcript, but then they slow down a little bit, limit protein production, or even completely stop protein production halfway. When you enable this type of approach, this also can trigger nonsense-mediated decay, and so you also could get a benefit of decreasing the RNA in addition to stopping the protein production. And so for today, I just want to show a brief example of kind of a therapeutic approach to slowing translation. And this is a therapeutic that was developed by Pfizer actually to treat cholesterol disease, but it's a really nice example of kind of how you can really fine-tune translation to have a therapeutic effect in treated disease. So Pfizer developed in 2017 a drug called PF846. This is a small molecule that enters the exit channel of a ribosome. So as ribosomes are translating, the growing chain has to exit from the ribosome complex so that the next amino acid can come in. For the majority of genes that are translated and proteins that are produced, this small molecule does nothing in the exit channel. But in a very select number of cases, it interacts with specific conformation of the protein that's coming out, and then it leaves it to get stuck in the exit channel, and this prevents the ribosome from moving. And so the ribosome then stalls, no more protein can be produced overall. And this is an extremely selective drug. It affects only 22 genes, with the target being PCSK9. So we also tested this in-house to identify stalling using our eRiboPro assay, where we treated HEC293 cells with PF846. We performed our eRiboPro assay, and then we identified stalls with a statistical test. I'm showing the result of that stall analysis here, where each dot is an individual transcript which has an individual stall. The location of the stall is along the x-axis, and the amount of the ribosomes that are accumulating is on the y-axis. Ribosomes do stall just transiently. It depends on what's happening in the cell, its cell culture conditions, and so on. So we're going to focus specifically on the ones highlighted in red, which are very strong stalls, and when this therapeutic was introduced. And the transcripts with the highest stalls are the ones that are reported to be induced with this therapeutic, such as PCSK9, PCPB2 as well. This is an abstraction. We can also look exactly what's happening with the dynamics of translation, which I'm showing here. This is a gene model. Each rectangle is an individual exon. And then these are the sequencing reads in a control, showing the amount of ribosomes at each exon. We get very nice equal coverage with equal translation occurring. And then when the therapeutic is present, we get a very strong stall. So all the ribosomes accumulate here, and then it can't proceed to create protein able to treat the disease by not producing PCSK9 at high levels. And importantly, this is selective to just the amount of translation. If we look at the RNA sequencing data, the amount of the RNA remains consistent. So this is a very selective, very fine-tuning control of protein production. So it's just a quick example of validation using ribosome profiling. But I did want to highlight that we do provide support for target discovery and drug validation on a number of dimensions for both small molecules, siRNAs, and ASOs through our lab services program at Eclipsebio, as well as the target discovery that I mentioned earlier. But I also did want to mention that we do provide support across the full spectrum from target discovery, drug design to drug validation through our eVerse portfolio of data sets to look at which are AI-ready data sets to support drug design, and then also our eMERGE platform to support RNA-based medicines such as cancer vaccines, as I mentioned earlier. And with that, I'm happy to end. We'll have questions at the end after Mike's presentation. Thank you very much.
Thanks so much, Wayne. So we'll be moving on to the second half of our talk with Dr. Mike Garbati from Active Motif, senior scientist here at Active Motif. And he'll be talking about a more upstream approach to help identify targets, to go after what Wayne just spoke about, and that's using single-cell multiome. So when you're ready, Mike.
Yep. Can't see my screen.
You are showing your screen.
I am. Oh, awesome.
But
awesome.
it's not full screen right now.
Let's go into presenter mode then.
There you go.
Okay.
Perfect.
Okay, great.
So,
Thanks.
and then before you begin, I'll just remind everybody, if you have questions for Wayne or during Mike's talk, you can enter them into the Q&A function at the bottom of your screen, and we'll try to address as many of those as we can at the end. So anytime you're ready, Mike.
All right. Thank you, Mason. And thank you so much, Wayne. So today I'm going to talk about a technique that we perform called single-cell multiome. And this is a single-cell assay that combines both open chromatin features and gene expression from the same cell. So next slide. So what is meant by single cell? Well, when we perform gene expression analysis, we are comparing that to bulk versus single cell here on this slide. So each cell type has its own gene expression profile, and that would be lost to averaging out if we didn't perform single cell. So single cell analysis, it reveals heterogeneity and subpopulation expression variability. So what that helps us to do is learn about different cell types that could be rare in a population. Some of them could be cancer stem cells or tumor cells. And if we only perform bulk analysis, we'd lose that heterogeneity. So why single cell is to identify subpopulations of cells with different epigenetic or transcriptomal profiles within the same sample. And this can eliminate the need for using FACS or magnetic sorting to isolate those cells. And of course, those sorting technologies are typically dependent on surface protein expression, whereas single cell can define a cell type by its gene expression and open chromatin features. We can use also this technology to identify drug responders within a population, or resistant cells, or tumor cells versus normal cells within a heterogeneous population. And our assay has a sensitivity to detect a population representing only 1%. So next, I'm going to go over the workflow. The most important aspect, or the most important step to getting a good single cell experiment, is to have good quality nuclei. In our lab, we're starting with two different sample types, either frozen cryopreserved cells that were viably frozen by our customers, or frozen tissue these tissues were harvested and flash frozen on liquid nitrogen. So with the cryopreserved cells, we want to do a slow dropwise thaw in media and measure viability. We're looking for highly viable cells that we'll then lyse and get good nuclei from. From the frozen tissue, though, we use a specific kit. It has filtration and debris removal to then end a lysis buffer that then allows us to count high-grade nuclei and grade the debris. Now, no sample is going to be perfect. We're looking for a high proportion of good nuclei with very little blebbing, clumping, and debris. And there are examples where there is some debris there, but it's acceptable, and you get a good data set from it. So next, what happens once you have good nuclei? Those nuclei are loaded into what's called the Chromium controller. What I'm showing now is the 10x platform. That's what I'll be covering. There are other competitor platforms, but the assay that we use in our lab is the 10x platform. So these nuclei are treated with TN5 transposase. This produces an open chromatin library. They are then met with 10x individual barcoded beads so that one bead meets one nucleus. 95% of the events are one bead, one nucleus. We then collect them. The individual barcodes are ligated to the attack fragments. We perform RT. Once again, the individual barcodes are ligated to the cDNAs. So next, still in one nucleus, we've got individual gene expression and open chromatin profiles with a common but unique barcode so that each can be traced back to the other from the same cell. So then after some amplification and library prep, we can then perform sequencing and analysis. So next I'll show you an example of some of that analysis. So the next few slides are going to be from my upcoming poster at Immunology 2025. In this example, we're looking at how Multiome identifies different cell types in blood. And in this case, it was peripheral blood mononuclear cells, PBMCs. Today, I'm going to focus on a specific cell type called monocytes and their progeny, the macrophages. And what we're going to do is go through a few different tissue types and demonstrate that we can find the same cell type in different tissue types. So here we've identified monocytes, and traditionally they're defined by CD14 expression, but that's an old designation. Now we can define them by all of the open chromatin and gene expression profiles and features that define them as a monocyte. These cells are in many different tissue types. Now they originate from the bone marrow. Most of them go to the blood, but they migrate to other organs. And so I've put arrows in front of the three tissue types that I'm going to focus on today. I've already shown you that you can identify monocytes in the blood, But we're also going to look at mouse brain and mouse heart and see that the same bone marrow-derived macrophages can make it to those different tissues as well. And we can use multiome to define those cell types. So here on the left, this is a merged data set of both the gene expression and open chromatin profiles from mouse brain. This population labeled micro 3, they are the microglia. And also in mouse heart, we can identify macrophages in the heart as well. So next, I'm going to show an example of one gene. If we just picked one gene and followed its gene expression in open chromatin profiles, can we see it in all three of those cell types? So I chose CSF1R because it's highly expressed in blood monocytes, and its promoter is open. So we can look at both of those aspects of CSF1R. And as you can see by the blue or purple dots, those are cells that have high expression of that gene. And then when we look at the ATAC features, we can see that in that same population, the monocytes that I've highlighted at the top, they also have open chromatin at that promoter. So what if we look at the other cell types that are actually in mouse? Do we see the same thing? And the answer is yes. So if we look at brain microglia, we can find that same gene, CSF1R. This is the mouse gene now, highly expressed in that population that is similar to the blood monocytes, and also similar to the human blood monocytes. Its promoter is open when we look at the single cell ATAC data set from the mouse brain. And similarly in mouse heart, while the ATAC is not as striking, there is still some open chromatin here in that mouse macrophage population that's in the heart. And it seems to be the only cell type in the heart that has high gene expression of that same gene that I've chosen to look at through all three of these different tissue types. So I chose that one. There's a list of many genes that are commonly highly expressed in human blood monocytes, mouse brain microglia, and mouse cardiac macrophages. So this is just showing something that you can do to compare different tissue types and the power of multiome to show you, given a specific gene or a specific promoter, where it's either highly expressed or highly open. So in the third phase of my talk, I'm gonna bring up an example of how multiome can define a cancer type. So this is a paper from INSERM in France. This group has long been characterizing hepatoblastoma, which is a type of liver cancer. And here they've performed single cell multiome on 6 different hepatoblastoma patients. And as we know with many cancer types, given a specific cancer type, you can choose 6 patients and see 6 different types of that cancer. So this group has dug deep into those differences and defined them. So firstly, they've merged these data sets into a single UMAP plot, both for on the left open chromatin and on the right gene expression. And because they know the features of hepatoblastoma tumor, they can define which which populations are which are tumor cells versus the normal cell populations. They can also define which patient had which population, and they know that based on the discovery of oncogenic mutations. So chromosome 11 P15. This is a known alteration. There's copy neutral loss of heterozygosity is a very well characterized alteration in hepatoblastoma. And they're showing that this exists in 5 out of the 6 patients. And there's one patient where there wasn't loss of heterozygosity at that chromosome 11. Another example of a feature of hepatoblastoma are beta-catenin mutations, and they can detect different mutations in this gene across the 6 different patients. And what they're showing here is that those 6 different patients have 6 different mutations. And one of those patients has 2 different mutations. So once again, it's one disease, but it's defined by several features. And if we just focus on the tumor cells and call them out by which patient had which cells, we can look in both the open chromatin regions and the gene expression to call out the different subtypes. Clearly, one of the patients has a mesenchymal subtype of hepatoblastoma, but the other 5 patients have a mixture of different subtypes, either liver progenitor or hepatocyte origin or both. So this is just another application where you can define different cell types based on different tumor cell types based on what cell of origin they came from. So in conclusion, single-cell multiome can provide both single-cell gene expression and single-cell open chromatin data from the same cell, and you can trace each library back to the same cell. It can be used to cluster different cell types within a sample and view highly expressed or highly open chromatin features within a cluster. It can also discover different cell types based on its gene expression and open chromatin similarities that define them as distinct cell types. Multiome can also be used to discover tumor cells, oncogenic mutations, and identify cancer subtypes, as illustrated by my one example that I presented here from last year's Nature Communications published by INSERM. And lastly, I'm going to end on this slide. We do this as a service in the group that I lead here at Active Motif. We perform single-cell RNA-Seq, single-cell ATAC-Seq, and the combination single-cell multiome. We were the first lab to be certified as service providers by 10x Genomics. And coming with that, we have a wide range of sample types that we've been successful with. So this is a kind of a short list. Actually, it's growing, and we continue to get many different samples in-house, and we've been very successful on these and new samples too. So with that, I'll conclude the talk here. Mason, do you want to take over and start questions?
Sounds good. Thank you so much, Mike. Again, everybody in the audience, if you have questions for either Wayne or Mike, go ahead and type them into the Q&A, and we'll try and get to them. So Wayne, the first question I have is for you, and it is, sorry, are you Yeah, you're there. Okay. Just checking. So Wayne, are ribosome stalls only caused by small molecule drugs?
Oh, no, they can actually be caused by a lot of different features. For example, it can be due to how the RNA itself is folding. That can lead to it to stall, modifications, protein binding, areas of therapeutics. There also have been designs using ASOs or shRNAs to actually trigger stalls to occur as well.
Great, thank you. The next question is also for you, Wayne. You talk about triggering NMD. Can you also see or confirm that using Eclipsebio's technology?
Yeah, so NMD is nonsense-mediated decay. So that is a situation that when ribosomes stall very strongly, the cell will recognize that something's going wrong, the protein's not folding correctly, or there's some other aspect, and so we'll start to degrade that RNA. And then with our technologies, we can actually see that. So eRiboPro is looking at translation as I had shown, but it also looks at the level of the RNA. And so we do statistical tests to see is a given gene actually changing the level of the transcripts that are present in addition to the translation. So we do provide information on both of those components.
Thank you. Next question. I am not sure if I fully understand it, but we may need to follow up. But is nuclear expression well correlated with cytoplasmic expression? I'm not sure.
I'll take that one.
Okay, great.
I was actually in the middle of typing a response into the Q&A.
Okay.
But I'll say what I was going to type. So the answer is yes. And on our website for single cell gene expression, I've posted 4 review articles. And these articles have the general consensus that the gene expression profile that you get from nuclear RNA-seq is very similar to the gene expression profile you get from whole cell. So I'm gonna so if you can see in the Q&A, I've posted an answer to that question. So if you click on that link and scroll about a third of the way down, there's a box on the right, has 4 different review articles. So yeah, admittedly, it is different, but it's similar enough. The consensus is that nuclear gene expression is a valid gene expression profile.
Thanks, Mike. Okay, I'm going to paste that into the chat too, just so make sure everybody can see that link. That was the last question I see in the Q&A at the moment. Maybe another one will pop in while we're wrapping up, but thanks everybody for joining. And if you have further questions for either Mike or Wayne or just in general, feel free to reach out to either company. You can reach Eclipse at eclipsebio.com or ActiveMotif at activemotif.com if you have any other questions or just want to take us up on any of our products or services. Thanks for joining, and you should be able to see this webinar posted on either of our sites as well in the near future. So thanks so much for joining, and have a great one.